Hello everyone and welcome to this video. In this particular video, we are going to talk about the amino acid as weak acid, bases and buffer. So amino acid act as a weak acid as well as base is because of the amino group and the carboxyl group present on it. So the amino group provide the basic group and the carboxyl group provide the acidic group. So presence of both make them a good buffer. So all of the amino acid have at least two tetrable protons and therefore they have two pKs, one at the alpha carboxyl group and another at the alpha amino group. So some amino acid have a third tetrable proton also. So in case of the aspartic acid, glutamic acid, the R group also contain the another carboxyl group, and in case of the arginine and the lysine group, the R group contains the amino group. So if we look at this particular model amino acid, which is alanine, at low pH, all of the tetrable proton are bound to amino acid. So as we increase the pH, so one of the proton is being released by the carboxyl group, and at this particular point the net charge on the amino acid is neutral. So at a low pH the net charge on amino acid is positive because here we have NH3 positive group and the rest are neutral. So at this particular point the amino acid is having a net positive charge. So as we increase the pH, the carboxyl group loses its proton, and it at this particular point the positive and the negative group are neutralizing each other. So at this particular point or the pH, the net charge on the amino acid is neutral or the zero. And as we continue increase the pH, so what happens is the NH3 group also loses its uh, proton, and at this particular point the net charge on the amino acid is negative. So this is how the amino acid work as a weak acid as well as base. So if we look at amino acid as weak acid, so all of the properties of amino acid in a protein and peptides are actually determined by the R group. In addition to that, the charges also play a important role because they ultimately affect the protein structure. So if a particular amino acid is having a net positive charge or net negative charge, so that will determine that they will be in the core of the protein or in the surface of the protein. If in general, if amino acid is having a positive or a negative charge, they are usually found at the surface of the protein, and thus it is important to know which group on peptide and protein will be protonated at certain pH. So for example, I have shown here the general amino acid, not shown actually, I have taken this picture. So if we look at the uh, basic formula for the amino acid, it is having a NH3, CCOH, H and the R group. So at low pH it is in a cationic form. So as we increase the pH, it will lose its proton, and at this particular point, it will be a neutral because there will be no negative charge. This is called deuterion. And as we continue increase the pH, it will lose another of its proton at the NH3 group, and it will come in the anionic form. So on net charge will be a negative charge. So the degree of dissociation of the proton actually depends on the pH of the solution. So now if we look at the titration curve of general amino acid, which is having a two tetrable protons. I am not considering here the aspartic acid or the lysine group, which is having a R group having a additional NH3 or the carboxyl group. So at the low pH, the moiety, so the here the NH3CrCOH or the bond group is more than the unbound group, or the here the acidic group is there than the its conjugate base. So you can say in vice versa because we have a NH3 also and the COH also. So it is a basically a buffer. So as we continue at this particular point, the A moiety is greater than the B, and as we continue increase the pH, so what happens is COH actually loses its proton. So we get a curve like this, and eventually it reaches the point where the all of these moiety, the bound tetrable proton moiety and the unbound moiety are equal. So this is a point where the net charge on the amino acid is zero. So this point is also called the isoelectric point. And as we continue increase the pH, so what happens is the NH3 also loses its proton, and at this particular moiety we have a more of the negative charge zero. So this is how the titration curve of amino acid look like. So this is the sigmoidal curve. So for the amino acid having a 3 tetrable proton, so there will be a 2 sigmoidal curve. So one will be like that, and another one point here, and there will be another shift here. So now coming to how do we calculate this isoelectric point where the net charge on amino acid is zero. So first, what is the isoelectric point? So isoelectric point, or the pi, is the pH at which total charge on an amino acid or protein molecule is zero. So how do we calculate? So first we need to draw out the complete ionization of a amino acid. So from the amino acid at low pH to the neutral to the net negative charge. Then we need to determine the net charge on each ionized form. Then we have to find the structure that has no net charge. Then we have to take the average of pKa that are around the structure with no net charge. So we will calculate the pKa of 1 and pK of 2 and divided it by 2. So for the case of the alanine, or which is having a 2 amino or so 2 tetrable protons, so it is simple, we just take the average of the both the pKa's. But in case of 3 tetrable group, it is cumbersome. So for example, here I have shown the 3 tetrable protons. So it is our aspartic acid moiety. So at low pH it is having a net positive charge. So as we increase the pH, so it will loses it proton, and it will have the net negative charge, because the NH3 and CO minus is actually balancing each other out. So at low pH the moiety is having a net positive charge, and after addition of the pH it is having a net zero charge, and as we continue increase the pH, it will lose another of its proton from the R group. So at this particular point the moiety has a net negative charge, but because it has a 3 titable group, so as we continue increase the pH, so it will lose another of its proton at the NH2, and the net charge on the aspartic acid will be the -2. So from the +1 it went on to the -2. So first what we need to do is we need to look for the moiety which is having a net zero charge. So we will took that moiety, and we'll take the pKa from the left and from the right, and we just take the average of it. So in this case pKa1 plus pKr by 2 will be the pi for this particular moiety. So I hope you understood the video. If you did, like, share and subscribe, and if you have any question, please put on the comment section and I will try to help you out. Thank you.